In Lecture B of Chapter 24, now we're going to look at those nephrons that we mentioned before and take a closer look at those both by anatomy and also what happens within it. So we know that the nephron is the basic structural unit within the kidneys. It's there to, of course, filter the blood. We can modify the fluid that is within the tube, so whatever was filtered out within the glomerulus and the renal corpuscle will find its way through this tube system that is known as the nephron. So notice there's two portions to it. You have the renal corpuscle that we said has the glomerulus in it, that also has the afferent and efferent arteriole, and then you have the renal tubule and the subdivisions that are part of it. So if you notice, coming out on the opposite side of the afferent and efferent arteriole, you have what we call the PCT, or proximal tubule here. So this textbook calls it proximal tubule. A lot of times it's referred to as PCT, proximal convoluted tubule. So if you notice, it does have quite a bit of turns and kinks, and it's much longer than another section that we'll talk about in 1 minute. So coming out of the renal corpuscle, the PCT or PT, proximal tubule, then you have the nephron loop itself. So if you notice, there's an area that's thick and there's an area that's thin. There's one side that is descending, the other side is ascending. So that's going to play a huge role with keeping the medulla salty. And then notice the DCT, distal convoluted tubule, or distal tubule as it's labeled here, is not as convoluted and it's not as long. It's much shorter than the PCT. And then several distal tubules will enter into another duct that is known as the collecting duct, and it will follow the renal pyramid all the way down to its base, empty into renal papillae, and then the renal papillae would empty into the minor calyx. It would empty into the major calyx, and then the urine, of course, would enter into the renal pelvis and exit out of the kidney in the ureter that we mentioned earlier. Okay, so looking first at the renal corpuscle. So the diagram there shows you that we said you have the simple squamous epithelium as the parietal layer. There's a capsular space, so that's the area that's going to collect the filtrate from the glomerular capillary itself. And then we mentioned that you have a arterial side where you have the afferent and efferent arteriole that enter and exit the renal corpuscle, and the opposite end is the PCT, or proximal tubule. And then of course you have podocytes that wrap around the capillary itself on its visceral layer, and again it's all about controlling the filtrate from the blood, delivering it to the tube system of the nephron. So again, the renal corpuscle's major responsibility is to filter the blood of toxins, electrolytes, ions, to control all those homeostatic functions that we mentioned. Notice that the glomerulus is extremely leaky, very much permeable to all of these substances. And I mentioned earlier that it's one of the capillaries that is under the highest of pressures within the body, so it's going to force the filtration from the blood into the filtrate. Where is it going to collect? It once was called Bowman's capsule, but now I told you the area around the glomerulus within the renal corpuscle, you have the glomerular capsule, but the capsular space is there to collect all that fluid and deliver it to the proximal tubule. So again, you have a sheath of epithelial tissue creating the glomerular capsule, but the space that is there is known as the capsular space. So make sure you underline that, and I'll also point it out in the laboratory section of the class. So the capsule itself, we said, also known as Bowman's capsule. It has an outer parietal layer, we said, made of simple squamous epithelium. The inner visceral layer has modified epithelial cells with podocytes, these little special foot extensions or processes, also known as pedicels, that will create glomerular capillaries at their filtration slits, so they can control how much filtrate and the substances that will enter and exit from the blood into the glomerular filtrate fluid. We said the capsular space was the hollow region in between the glomerulus itself and the glomerular capsule outer layer. So we said it's continuous with the renal tubule lumen of the proximal convoluted tubule or the proximal tubule. So its primary responsibility, you need to make sure you understand that the capsular space is what collects the filtrate once it's been filtered from the blood using the glomerulus, which is a specialized capillary under high pressure but is very leaky, so that we said the filtering blood is made possible. Then, of course, I've mentioned several times that that fluid that is the waste products, it's a very high content of fluid, around 160 to 180 liters of fluid every day. And of course, we don't void that, but that filtrate is formed. So the filtered fluid with waste, metabolic waste, and with those different ions and electrolytes, glucose, amino acids, those types of things, are filtered from the blood and enter into the lumen of the renal corpuscle there at the renal tubule. And the diagram is the one from our lab model. You can see the afferent arteriole much larger, much thicker than the efferent arteriole as it exits out. Notice it shows you the glomerulus, which is the capillary itself, Bowman's capsule, also known as the glomerular capsule now, and the capsular space that allows the fluid that has been filtered to enter into the PCT. The nephron itself, we said, is now new filtrate is finding its way within the renal tubule. It can be modified for during three processes for the entire length of the nephron. So both structurally and functionally, there's three distinct regions I told you about. You have the PCT or proximal tubule, the nephron loop itself, and the distal convoluted tubule or distal tubule, also known as DCT. And then we said that those would empty, several of those distal tubules would empty into the collecting duct. So starting with the proximal convoluted tubule or the proximal tubule, I mentioned earlier it is the longest and also the first segment leading out of the renal corpuscle. It's very highly convoluted, meaning twisted about. Notice that you have simple cuboidal epithelial cells. So simple epithelial cells, cuboidal, is designed with the microvilli so that you can have all of that filtration that happens. So brush border microvilli and the simple cuboidal epithelium that is there. So notice, remember, microvilli increase the surface area and the brush border greatly increases that so that filtration can happen at a pretty fast rate. The nephron loop, once known as the loop of Henle, is the next segment, and I told you you have two parts to it. You have the descending part and then the ascending side. So the only part of the nephron that will actually enter in and dip into the renal medulla area is the nephron. So that will come back up when we talk about the countercurrent multiplier. So it consists, we said, of the descending side and the ascending side. The descending side, of course, is known because most of it is thin, and we'll see with simple squamous epithelial cells, you're going to allow water to move very readily through the descending side and the thin portion of it, the thin descending limb. Then it's going to travel downward toward the medulla. Notice on the opposite side you have the ascending limb, so it makes a 180-degree turn and then begins to ascend up, but this time the histology changes and you have simple cuboidal epithelial cells that are part of the thick ascending limb. So this is going to allow your electrolytes to exchange with the medulla, and it's going to keep the medulla salty so that as the collecting duct dips down through the medulla area, it being a salty environment, it's going to draw a lot of the water that's left over from the collecting duct back into the blood, and we said the vasa recta would be the capillary that would absorb it in the medulla, and it's going to concentrate the urine and create what we call a hypertonic urine later on. Hypertonic, there's more solute than there is water. So we said that the nephrons that have a thin ascending limb, they begin to bend at a region, and that region is, again, because of the histology, because you have the simple squamous epithelium. But the thicker cuboidal cells are found upstream as the fluid begins to arise back up. The DCT, or distal tubule, is less convoluted. It's not as lengthwise as the proximal. It is the last portion of the nephron before the fluid now enters into the collecting duct, and then it'll be called urine. So we've gone from glomerular filtrate, which is the fluid filtered from the glomerulus, to the tubular fluid, which we're going to modify by either absorbing things back into the body or excreting things from the body. And then in the collecting duct, now we have urine. So most of the DCT is simple cuboidal epithelium, but they do not have a brush border. So there's very little change in the fluid once it reaches the distal tubule. How is this all regulated? Well, you have an apparatus that's known as the JG apparatus, or juxtaglomerular apparatus. So where you have the transition between the ascending limb and the distal tubule, notice in the diagram you've got the glomerulus, you have the DCT that comes very closely there near the afferent arteriole. So these, this area around all of these nephrons have this apparatus. So notice it consists of a couple of different things. You have the macula densa, which are modified smooth muscle cells. They're also known as JG cells. Sorry. The juxtaglomerular apparatus regulates both blood pressure and GFR, which is the glomerular filtration rate, how much of the filtrate from the blood. The collecting system, you have further modification, we said, as it travels through that. So there's several distal tubules that go into the cortical collecting duct. Then you have the medullary collecting duct as it's dipping down into the salty medulla. Then we said that they will merge into a pair of, or not a pair, but a set of ducts that are known as the papillary ducts at the very end, and then those empty into a minor calyx. And we said that once it enters into the papillary duct, it's now known as urine, not filtrate, because there's no more changing that's going on except more absorption of water. And then, of course, the minor calyx that is at the very base of every renal pyramid. There's a couple of different types of nephrons. We said you have the cortex region and the medulla region. So the cortical nephrons are the majority that are found in the outer portion of the kidney. They have very short nephron loops, and they're associated with the peritubular capillaries. The juxtamedullary nephrons, these are the ones that dip deep into the medulla, and we said that they're associated with the vasa recta. And this, of course, is what we just said. So the peritubular capillary are associated with the cortex nephrons, and the vasa recta with the medulla or medullary nephrons. So notice their capillary structure and their function is to control both the volume of urine and also the concentration of the urine. So if we overdrink with plenty of water, of course, that water is going to be emptied out of the kidneys, and you would have hypertonic urine instead of hypotonic urine instead of hypertonic urine. It would be mostly water. So in this section, hopefully I did an okay job of trying to explain both the structure and the function of the nephron itself. Remember, it is the functional unit for the kidneys. It consists of a renal corpuscle and the renal tubules. The corpuscle is designed with the glomerulus to filter the blood with high filtration rates. The renal tubules are all about reabsorbing or resecreting substances into the fluid. We said the JG apparatus, or the juxtaglomerular apparatus, and the collecting system work together to concentrate the urine. We talked about the pathway of the filtrate and also the differences between the two types of nephrons.